All right, let's start. It's my pleasure to introduce today's speaker. It's her PhD exit seminar. It's Christy and she's going to talk to you about her work on methylmercury in California rice ecosystems.
Hi, thank you everybody for coming. So today I'm going to talk a bit. First, I'm going to tell you a little bit about what methylmercury is and why it's even an issue in California rice. Then I'll go over some of my overarching research questions, and then I'll talk about each of the 3 studies that I did for my PhD. So first, looking at methylmercury exported from rice fields, taking a big picture view at the watershed scale. Then zooming down and looking at it from the field scale, and then trying out a management practice to see if it would help address some of the issues related to methylmercury in rice, and then give a few of the conclusions from the research. So, what is methylmercury? Most likely, you guys may not have heard of methylmercury, but how many of you heard of the issue of fish having a lot of mercury? Yeah, all right. And turns out that the kind of mercury that's in the fish is methylmercury. It's not inorganic mercury, it's this mercury that's bound to carbon. And methylmercury is produced by these bacteria that live in flooded soils. So they're sulfate-reducing bacteria and iron-reducing bacteria. And they take inorganic mercury and they convert it to methylmercury. And that methylmercury bioaccumulates. And so fish that are at higher trophic levels have higher concentrations of mercury. So in rice fields, there's the perfect conditions for this to happen. Rice is grown with a continuous flood in most cases, and it produces a lot of carbon, which can increase the activity of microbes in the soil. So production of methylmercury in these rice fields can lead to a couple of issues, such as the methylmercury can accumulate in rice grain, which is a problem for human health, especially in areas where people eat a lot of rice in their diet. It can also impact wildlife that live in the fields, and it can also impact wildlife that are downstream. And then in places where people eat the fish that are downstream, it can also impact human health. And in California, we have about 200,000 hectares of rice in the Sacramento Valley. So that's the yellow area on the map. And there's also a history of mining. So mercury was mined in the Coast Mountains on the western side of the valley. And that mercury was taken to the Sierra Nevada and used to extract gold. So in both those areas, a lot of mercury was released to the environment. And downstream of the Sacramento Valley, in the Sacramento-San Joaquin Delta, is this kind of bluish-green area here. There are high levels of mercury. Wildlife in the area have found to have, fish have high levels such that we're worried about human health. Birds have reduced reproductive success because of this. And so there are regulatory actions that are being put together to try to limit how much methylmercury is coming into this system from the Sacramento Valley. And so we wanted to know, well, we know that there's rice, these flooded ecosystems. We know that there's mercury around, so how much methylmercury is coming out of rice fields? So I had a couple overarching questions. First is, how big of a problem is this? When and where is it a problem? So if we know when and where it's a problem, we can start to figure out how to manage it better by targeting those times. And what are some management practices that would work to reduce the impacts of methylmercury? And I started off looking at this from the watershed scale, the whole Sacramento Valley. And I used historical data. So the thing about mercury is that it's extremely expensive to analyze a single sample for methylmercury, about $300 for one sample. Luckily, there was all this data available, and I had actually 5 data sets, and they spanned a period of about 10 years. And so the yellow areas win each data set, the time period for each data set. And they covered different numbers of sites, and had a different number of samples. And so I compiled all these, and with the objective of investigating seasonal and spatial patterns in the data, comparing methylmercury concentrations from rice drainage water to concentrations in irrigation water, and then trying to estimate the total amount of methylmercury that's coming from rice fields. So in theory, it's a nice, tidy study where we have a river, and irrigation water is diverted out of the river in the upstream area. That's used to irrigate rice. And in those rice fields, theoretically, mercury is converted to methylmercury. And then drainage water from the rice fields is collected by agricultural drains and returned to the river. And then there's this downstream area where river water and rice drainage water are mixed together. In real life, it's a little bit more complicated than that. So here's a map of the valley again. Brown is rice area. The green is wetlands, and these are also places where methylmercury can be produced. So there are 2 sources of irrigation water. We've got the Sacramento River and then the Feather River over to the east. And those are both used to irrigate rice. And there's been a lot of manipulation of how water flows in the valley. So irrigation water is diverted out. You can see 2 main irrigation canals, the black kind of on the top over there. Then drainage water is collected and returned to the river. And we also have a bunch of flood control structures that carry extra water, especially during the winter. If any of you have driven across to Sacramento lately, you've seen some of those in action. So that's the yellow bypass right here. So all this makes it a little bit more complicated. But what I did is I took, so all the points on there are the sites where data was collected, and I grouped them by their location with respect to rice drainage water. So the blue points are, I'm calling upstream Sacramento. So these are on the Sacramento River, but upstream of areas where rice drainage water is coming in. So they represent irrigation water and water that hasn't been impacted by rice. The purple is the same thing, but on the Feather River. The 2 green points are 2 major agricultural drainage canals that enter the river. And then orange is downstream of all of that. So orange points are a mixture of river water from the Sacramento, river water from the Feather, and agricultural drainage water. So we used since not all of the sites were measured in every year, we had to kind of use a linear mixed effects model to account for that. So we had a random effect for site and year. So this just kind of tells the model that not everything was measured in every year. And then we had fixed effects, which is what we're testing for location, which is upstream Sacramento, upstream Feather, agriculture, or downstream, and season. And to decide on the seasons, a main question of mine was, when is methylmercury high and when is it low? Rather than, is it high in the growing season or is it high in the fallow season? So what I did is I took all 10 years of data and I plotted them on top of each other here. So all January is on over there, all December is over here. And you can see that there's kind of a low period in the late summer and early fall. So I called that it's May through October, and then it was November through May as my 2 seasons. And you can see the low period kind of lines up with the rice growing season, but not quite. So this is the results of the mixed effects model. These are modeled least squared means. So they kind of account for the differences between years and differences between sites. So all those multiple points that were upstream Sacramento kind of all got mushed together. And so the black is June through October. And you can see there's not really any significant difference in concentration during that period. So upstream irrigation water wasn't really significantly different from agricultural drainage water or downstream. But during November through May, there were elevated concentrations in agricultural drainage and elevated concentrations downstream. And that was a significant difference. All right, so next I wanted to calculate how much methylmercury is coming from the rice fields. And I did this using loads. So a load is when you take the concentration that's in the water in a river and you multiply it by the amount of water, the volume that's going downstream. So that gives you a mass that moves down the river over a certain period of time. And you can, change what period of time you're looking at. And the difficult thing about this is since it was historical data, there are a number of gaps. So I didn't have flow for the entire year for one of the agricultural drainages. Some of the water's diverted for irrigation and for flood control, and the downstream area had limited data. It was only available for 2 data sets. But some of the results are here. And so the colors in the bars correspond to the points on the graph. So blue and purple are upstream Sacramento and upstream Feather. The green is our 2 agricultural drainage sites. And this is only the period from June through October. So we would expect that the 2 upstream sites and the agricultural sites should add together to be about the same as the downstream, which didn't quite work out. And so each of the groups of bars is all represents an average of the entire 10-year period. And then we have separate data sets. And only 2 of those data sets, data set 1, data set 5, had data for the downstream. But overall, the agricultural drainages were about 13% of the methylmercury in the river. So some, but not as much as we would have expected. So another thing that I wanted to do is try to see how do these concentrations compare with previous studies. So there were a couple studies that were done in the Delta, and so the 2 on the left, the Yolo Bypass and the Cosumnes River Preserve, were field scale studies looking at methylmercury going in and out of rice fields at the field scale. And I show the concentration of irrigation water and the concentration of drainage water. And for comparison, we have the Sacramento Valley. So this is the data I was looking at. And there's a lot of spread in the data, but it came out to about a 10-fold difference in overall concentration between the valley scale study up in the Sacramento Valley and the field scale studies in the Delta. And for reference, there's a regulatory kind of goal. It says TMDL goal of 0.06 nanograms per liter. They're trying to get concentrations down to there, and these are the concentrations we've measured. So there's some work to do as far as that. But we wanted to figure out why is there a difference between the field scale studies and the watershed scale study. So it could be the studies are looking at it from a different scale, right? Field scale versus watershed scale. Maybe there's something going on at the watershed scale that's changing the results. Or maybe there's a difference between the fields in the Sacramento Valley and the fields in the Delta. So next, I conducted field scale studies in the Sacramento Valley. So these dots down here in the delta are the two studies, field scale studies that were done previously. And the dots up above are the locations of the fields that I looked at. And these are commercial fields managed by commercial rice growers. And I measured methylmercury and total mercury concentrations in the irrigation water going into the field, drainage water leaving the field, as well as soil and within the rice plants. And one of the first things that jumped out about these fields is that the total mercury concentration, so THG, in the soil was quite a bit lower in the fields I was looking at compared to the fields down in the delta. So the fields in the delta are around 300 nanograms per gram of total mercury. And so this is model results from the surface water data from the field. So it's a two-way ANOVA. I compared a categorical variable for inlet, outlet during the growing season and outlet during the fallow season, and then the field. And the reason I didn't have a separate value for inlet in between growing season and fallow season is that there were only one or two data points for the inlet during the fallow season, and they were pretty much the same. So when the fields are flooded in the fallow season, the growers will flood them up very quickly, so the water wasn't flowing that long, so we only had time to take a couple samples. But if you look at the top panel, you can see that methylmercury concentrations in the inlet water and during the growing season in the outlet water were not significantly different, but the fallow season concentrations were much higher. So this is similar to what we saw at the watershed scale. Higher concentrations in drainage water in the fallow season, but water was similar to the irrigation water during the growing season. Pretty much a similar result for total mercury, except drainage water actually had significantly lower concentrations than irrigation water. And both fields, there was a difference between the fields that was consistent with a difference in total mercury, so in the soil. So YOLO field had higher total mercury in the soil, higher methylmercury, and higher total mercury in the water. And Butte field was lower on all of those. So a metric that we use when looking at methylmercury is often the percent methylmercury. So this is methylmercury divided by total mercury. And both fields were behaving similarly. So this kind of gives you an idea of how much methylmercury is being produced and whether or not there's methylmercury production. So we can see that during the growing season, the outlet had elevated percent methylmercury. So there's some sign of methylmercury production going on there. And in comparison to the previous studies, so on the far left, we have the two studies from the Delta, then the Sacramento Valley study, and then the two fields that I looked at on the right. And the fields that I looked at were fairly similar, somewhat, to the Sacramento Valley, in that the concentrations were lower than the ones that were seen in the Delta. And note, this is a log scale axis, so there's actually, if it wasn't a log scale axis, you couldn't see these. So it suggests that in the Sacramento Valley, there's actually less mercury contamination than we originally thought. All right, so then I looked at methylmercury and total mercury imports and exports in surface water. So all these numbers are it's a load, it's in nanograms per meter squared of the field. and this is an import or export for the entire season. So during the growing season, the irrigation water that came into the field already had a decent amount of methyl and total mercury for both fields, and the amount that was exported in drainage water was actually much smaller. So the fields were acting as what we call a sink for methyl and total mercury. A bunch came in and it didn't come out, at least not during the growing season. So that's why we've got a negative number for export minus import. The opposite thing happened during the fallow season, where not very much methylmercury or total mercury was put into the field in irrigation water, but quite a lot more came out. So kind of 2 to 10-ish fold more came out. So we have the fields acting as a methylmercury and total mercury source during that time. So it seems like the fields tend to be storing methylmercury and total mercury. It stores up during the growing season and it's released during the fallow season. And so this is annual imports and exports. So both fields actually accumulated total mercury. If you look at just the surface water budget, they were net sinks for total mercury, but one was a net sink for methylmercury and the other was a net source for methylmercury, but they were both pretty close to zero. And then if we look at the rice crop, so any methylmercury or total mercury that was in the rice grain is going to be exported from the field. And these are in the same units as the water, whereas the straw is going to be incorporated into the soil, and so you have mercury going from the soil to the rice plants and then returning to the soil with the straw. And these values, as you can see, are fairly large in comparison. They're a substantial and similar amount compared to the surface water budget. So this is an important aspect of mercury cycling in these fields. So overall, we found that the fields I looked at, the patterns of mercury import and export were very similar to what was seen in the YOLO bypass and the delta, so those previous studies. But the values, the amounts of methylmercury and their concentrations were overall lower. So both fields had a lot of methylmercury going into the field during the growing season, not very much export. The opposite during the fallow season. And then the movement of the amounts that were in grain were comparable to the amounts in water. All right, so now that we had an idea of how the relative risks of methylmercury in rice in the Sacramento Valley, we wanted to test out a management practice. And one management practice that the lab has been looking at is alternate wetting and drying. So this is water management, and so in normal rice production, you would flood the field and plant the rice, and the field would stay flooded until a couple weeks before harvest. In alternate wetting and drying, you, at a certain point in the growing season, you turn off the irrigation water and let the water subside, so through evaporation and transpiration, until the soil gets to some predetermined level of dryness. And then you turn on the irrigation water and flood it back up again. And you can do that a couple of times. And a couple previous studies have found a number of benefits for this practice. It reduces water use. It reduces methane emissions. So methane is produced when you have lots of carbon in a flooded soil over a long period of time. So when you dry it out, you stop the methane production. Reduced arsenic accumulation in the rice grain. But there's it's a little risky because rice is a plant that likes to have plenty of water. So if you dry the field, you risk stressing out the rice plant. You can also increase weed pressure if it's, so water is an important weed management tool in these systems, and have the potential for producing nitrous oxide, which is another more potent greenhouse gas. So you can if you don't do it right, you counteract the benefit of getting reduced methane emissions. So I wanted to know how this would affect methylmercury. So the bacteria and other microbes that produce methylmercury need a couple of things to do that. They have to have anoxic conditions. So if there's too much oxygen around, they get outcompeted by other bacteria. And in some cases, they're poisoned by oxygen. They need a mercury that is mercury 2, so it's in the 2+ redox state that is available for methylation. And they need electron acceptors, which they use in place of oxygen, so sulfate or ferric iron, iron 3. And they also need electron donors, which isn't something that would be changed by this practice, I don't think. And so if we think of ecosystems along a continuum where we go from flooded, that is, say, a sediment at the bottom of a lake that's flooded for year after year after year, all the way to soil on the top of a hill that never actually stays saturated for much time at all, say in a desert. You'll have anoxic conditions and availability of electron acceptors are going to be negatively correlated. So in the sediment that's at the bottom of the lake, that's going to stay anoxic all the time. But the bacteria that are there will be using up those electron acceptors. So they take sulfate and convert it to sulfide, and they can't use it anymore. And ferric iron and convert it to ferrous iron, or iron 2. Whenever a sediment gets dried, those electron acceptors, so the sulfide and the ferrous iron, get converted back to the form that the bacteria can use. So when you come to dry soil, there's plenty of electron acceptors, but there's no anoxic conditions. There's too much oxygen. So aerobic dry soils have very little methylmercury if they haven't been flooded in the past. Soils that are, say, in a permanent wetland have some methylmercury production, but in comparison, tend to have somewhat lower levels of methylmercury compared to a wetland that is seasonal. So it goes from flooded to dry to flooded again. So that cycling between, which you see in a rice field, between flooded and dry helps make sure there's plenty of electron acceptors around. But there's also large periods of anoxic conditions where the bacteria can be active and producing methylmercury. So I looked at this as two alternative hypotheses. First, that it depends on what these bacteria are most limited by. Do they not have enough electron acceptors, or do they not have enough time to methylate the mercury because the soil's oxidized, not anaerobic enough? So the hypothesis 1 is that they are limited by electron acceptors. And if we go from continuously flooded rice, CF, to alternate wetting and drying, which means the soil gets dried more often, then we'd have more regeneration of electron acceptors. And if that's what limits the bacteria, then we'll have more production of methylmercury. But if, say, they've got plenty of electron acceptors, but they're limited by the amount of time that the soil is anaerobic, so they're limited by actually being able to be active, then AWD will make it so that there is less time that the soil is anaerobic and less time that they can be active and less methylmercury production. So we compared two treatments. AWD, which had two drying events so we dried down the soil twice, and we dried it until it reached a 35% volumetric water content. So this is about 7 to 8 days of being below saturation. And continuously flooded, which is a conventional rice production in California. And in California, during the fallow season, the straw is tilled into the soil and the fields are flooded. So both plots were managed that way, and we continued sampling throughout the fallow season to see see if the impacts continued. All right, so this was a randomized complete block design. We had 0.2 hectare plots with 3 replicates, and it was conducted at the Rice Experiment Station in Butte County, California. And the treatments were actually applied from 2012 all the way through 2015 without re-randomizing the plot. So we had 4 years of this happening year after year in these plots. But I started collecting samples in harvest of 2014 and then throughout the year in 2015. And a couple of the plots I'll be showing you will look like this. On the x-axis, I have days after planting. So 0 is the day that the plots were flooded and the seed was spread. And so the white areas are times when both treatments, AWD and CF, in both treatments the soil was dry. The dark gray means both the treatments were flooded. And then the light gray stripes represent times when there were the drying events. So the AWD plots were drained and the CF plots were still flooded. And so on the y-axis here, I have soil water content. And the black line is AWD. You can see that as soon as we start the drying event, this soil water content decreased. We went a little bit past 35% on one of the drying events, but CF stayed at saturation. All right, so the panel on the left has the one sample from harvest in 2014, and you should compare that sample to the one here with the same dotted lines. That's harvest in 2015. And first thing is total mercury in the soil. This is soil data, which I should have put on there. Total mercury in the soil did not differ between the treatments, and it didn't change throughout the course of the season. Whereas methylmercury, there were significant difference in methylmercury concentration toward the end of the growing season, so just before harvest to a little bit after harvest. Those values were significantly different between the treatments. And then toward the end of the fallow season, you can see that the differences between the treatments, when they're not treated differently, start to go away. So to get an understanding of how this management practice is affecting the soil chemistry, we measured iron speciation. So some of the bacteria that methylate mercury are iron-reducing bacteria. That means they take iron 3, ferric iron, and reduce it to ferrous iron, iron 2. So the top panel is ferrous iron. So that's the waste product of these bacteria. They can't use it, but it means that iron reduction has occurred. And then iron 3 is the electron acceptor that they can use. So you can see that at the beginning of the growing season, at just before day 0, there was not very much iron 2 and a lot of iron 3. So this is the, yeah. And when the plots were flooded, these get converted to, you have a lot more iron 2 and a lot less iron 3. So iron reduction has occurred. This could be, we can't distinguish exactly between whether it's microbial or abiotic, but this tells us that the soil is changing from an oxidized state to a more anaerobic reduced state. And if you look at the CF plots, once they get later in the season, they pretty much stay reduced. So they've got plenty of iron 2, basically 0 iron 3. But when the first drying event happens, the AWD plot goes right back to where it started at the beginning of the season, with plenty of iron 3 and not very much iron 2. So this shows that the treatment is working how we expected it to. It's making the soil more oxidized, so more aerobic conditions, less anaerobic conditions, and it's increasing the amount of electron acceptors that are available potentially for methylmercury reducing or methylating bacteria. Right. So this combined with the decrease in methylmercury shows that treatment was working how expected, and we were more consistent with hypothesis 2. And so the results from the soil were reflected in the surface water in the field. So I have the 2 treatments and then the growing and fallow season for methylmercury and total mercury. And again, these are modeled results. So for methylmercury, there was a significant reduction in concentration in surface water, and it wasn't significant for total mercury. And like the previous studies that I showed you, there were significantly higher concentrations of both methylmercury and total mercury during the fallow season than in the growing season. So we also saw it in the rice grain. So these are data collected at harvest 2015, about a month before harvest, harvest 2014, a month before harvest in 2015, and harvest 2015. And this is a significant reduction in both methylmercury and total mercury in the rice grain. But overall, these concentrations for rice grain are quite low. So there's a review that looked at a wide variety of papers that measured methylmercury and total mercury in rice grain, and these are the ranges that they saw, and compared to my data are right at the bottom of those ranges. Right, and since we're looking at agricultural production, we want this management practice to potentially be used by growers, we have to look at yield. And over the 4 years, there wasn't a significant difference in yield between the 2 treatments. In fact, if anything, it sort of alternated from year to year. So this is very promising. So it means you can get the benefit of reducing the amount of methylmercury without compromising your yield. All right, so overall we saw a reduction in methylmercury, and this occurred in the soil, in the rice grain, and in the rice surface water. And so that supported hypothesis 2, that when AWD reduced the amount of time that the soil was anaerobic, it reduced the amount of time that the bacteria could be active. And so even though there were more electron acceptors around, they couldn't use them. All right. So to conclude, methylmercury concentrations in rice drainage water were higher in the fallow season and lower during the growing season. And I saw this in all 3 studies. The rice fields tend to be net importers of methylmercury during the growing season, so sinks, and net exporters during the fallow season. So together, these 2 results suggest that we want to be able to target the fallow season to, if we want to reduce the amount of methylmercury coming out of these rice fields, the fallow season is when we need to look. Rice grain from the Sacramento Valley has fairly low methylmercury concentrations. And this was true for both studies that I looked at the rice grain for. And finally, alternate wetting and drying looks like it's an effective way to reduce methylmercury concentrations in the soil, water, and rice grain. All right, and with that, there are a lot of people that have helped me along the way. So I'd like to thank my major professor, Bruce Lindquist. I had really great co-authors and other people who advised me along the way. So Lisa and Stephen are here. Ken Tate, Mark, Jacob. So they all gave me great suggestions on my papers and have helped me out a lot. Cesar Abernilla and other members of the lab were always there to help out for field work when I need it. And Ray Stogsdill, who's up at the Rice Experiment Station, is always around helping out, making sure that the field experiments were running smoothly. So I wasn't able to analyze the methylmercury and total mercury samples here. We sent them to a lab at the U.S. Geological Survey. And there's a lot of people at that lab who put in a lot of work to make sure that I had high quality data. So Evangelos Koukouros, Michelle Arias, Lee Q, Jennifer Aggie, and Melissa Meridian. I had a number of people who reviewed my manuscripts and gave me great comments. So Sanjay Parikh, Tamara Kraus, and Charlie Alpers. Finally, George Rodriguez and Aaron Thompson and Sanjay helped out by loaning equipment and giving me recommendations for how to do the iron speciation analysis. And finally, I'd like to thank Lisa Brown because she's always there helping out, making sure that everybody's doing the stuff that they need to do to graduate and have funding. And my funding sources. So I had funding from the Rice Research Board, UCANR, the JASTRO Award, Plant Sciences, and the Golden Fellowship. All right. So if you have any questions, I'd be happy to take them.
Do you think that the mercury that's in the field now is metal mercury is going to be enough mercury later on in some other way?
Yeah, so like the total mercury?
Yeah.
Yeah, possibly. So that's going to probably end up in aquatic environments downstream all the way to the delta, possibly eventually after many years farther out to the ocean, where it could be methylated. And there's a lot of debate going on kind of around the delta about whether or not they should be focusing on mercury, total mercury coming in from outside, or methylmercury production within the delta. So there's some, the mercury doesn't always have the same ability to be methylated, and it depends on its kind of chemical form. And I think there's still a lot of research going on to try to figure out how to tell exactly which of that, what is most likely to be methylated.
So one of the impressions that I had from your talk is the idea that, well, maybe it's not that important in rice.
Yeah.
Right?
Not that important in rice in California.
In California, yes. Thank you. But every time I go to the bypass, the yellow bypass there. I see a lot of birds and I see a lot of animals there, feeding on whatever.
Mm-hmm.
So I'm assuming that the methylmercury is, that place is a sink. Those birds are taking up some of the methylmercury during the season or something that's going on.
Yeah.
Should we be concerned about wildlife and contamination and how that?
Yeah, so kind of one question about these rice fields is, should we be concerned about what's coming out the bottom of the field, or should we be concerned about what's actually in the field affecting the wildlife there? And it seems like there is some accumulation of methylmercury in the field, but also when fields are sinks for methylmercury, that could mean two things. It could be storage, or it could be methylmercury degradation. So it could be that the methylmercury in the field is actually getting degraded. It could be stored in the soil. It doesn't seem to be in a super high concentration in the surface water, at least during the growing season. During the fallow season, it's much higher in the surface water. So yes, it is a concern. And I think some of the ongoing research will be looking at kind of trying to figure out how much of the mercury that's in the rice surface water is in a dissolved form versus bound to particles. So that will kind of determine somewhat whether or not it's more or less likely to be bioaccumulated.
Hey, Christy.
I've got somebody on the video conference. Yes.
Yeah, this is Mark. Actually, a couple of quick things. Could you recap the questions if more come in? Because it's hard to hear those and stand closer to your mic.
Okay.
But I have one quick observation that I don't know if we caught in the paper, the field study paper where you were comparing YOLO and Butte.
Mm-hmm.
And as you had that table up where you looked at export minus import, and you did it for the growing season and you did it for the fallow season, because I was eyeballing those numbers. If you add that up for the full year, sort of the totality of all the months, those numbers sure look like net sinks overall. And I don't recall if we brought, if number one, if I got that right, and number two, if that's true, if we really brought that out in your paper, and if it is true, that's actually a pretty novel observation. Yeah, there you go. So is that the one I was thinking of?
Yeah. So
The one before, well, let's see.
so this is the sum of the one before, which so
Was
both fields were net sinks for total mercury, and one was a sink for methylmercury and one was a source.
Actually, there was another slide where you had it broken down by season. That's the one I'm thinking of.
Yeah.
Right. So, yeah, you have export minus import for growing season, and then the last row is export minus import for the fallow season.
Mm-hmm.
So, if you add those two seasons together, you still get negative numbers for Butte, it looks like.
Mm-hmm.
Positive number for YOLO, I see.
Yeah.
And a negative number for total for YOLO.
Yeah.
Okay, and I'm trying to recall, I don't know if Lisa Marie is in right now in the audience,
She's
if we
there.
did a real similar kind of comparison ourselves for the systems further downstream, the YOLO bypass type fields, in terms of the whole year, adding it all together or not. But the fact that even in the Butte, you actually saw a net negative methylmercury for the whole year combined, may say something about these systems that are not really in zones of really high mercury, because you're further up in the Sacramento Valley, compared to the more impacted portions of the YOLO bypass downstream.
Mm-hmm. Yeah, I think we probably need a little more data to say that for sure.
Yeah.
Lisa has a question.
It's a global question. So there's mercury everywhere in the atmosphere. It's a global pollutant. There's rice everywhere in the world. I'm just curious how you think your study might be applicable or not to the global issues of mercury and rice.
Okay, so Lisa's asking how this study might be applicable globally. So I think one thing that the field study, first of all, mercury in the atmosphere, we've got mercury deposition around the globe. So once it goes into the atmosphere, it tends to stay there for a long time and it gets spread all over. So there's a lot of places where they weren't initially very high mercury, but they are receiving mercury deposition. And I think there are definitely rice growing areas that are highly contaminated, and there's been a lot of research done there. But a lot of other areas that are not very contaminated, but receiving some mercury. And I think by looking at fields that had low total mercury in the soil, those results would be applicable to places around the globe that also are growing rice, but in somewhat lower mercury conditions. I also think that, it's useful to know that from the watershed scale, it looks like what we're seeing at the watershed scale is basically what we're seeing at the field scale, but scaled up. We didn't see any evidence of anything else going on. We didn't see any, since the concentrations were consistent with the field scale studies in that area. So that's useful for other rice growing watersheds. And I think the alternate wetting and drying is a management practice that could be implemented around the world to reduce methylmercury concentrations. It should probably be tested in more contaminated areas. So, and by contaminated areas, there's the places where this is really a problem are areas in China, and the soil total mercury is about 1,000-fold or more higher than I was looking at. So they really need something to help control methylmercury levels in the grain. And this management practice would probably work, though it should probably be tested in those areas.
Would they have that much control in those systems to like turn the irrigation on and off?
That's a good question. I think in some places they end up having to do that just by the way that, because they don't have quite as much control, so that might be a thing. But yeah, a barrier to using this alternate wetting and drying is making it actually work at the field scale, and that's going to have to be tested. And in California, we're trying it in the field this year. The lab is, but seeing whether or not that works in other areas with less control over the irrigation and stuff will require some work.
All right, we'll have to ask Christy questions after the seminar ends. We'll let students go. Thank you very much.